I'm going to tell you a story tonight, and it's the story of an endangered shrub that's highly restricted in its distribution. It's a story of a dam retrofit for public safety, a high bar set by a habitat conservation plan, really cool research studies, and a series of collaborative partnerships. So this is a map of Santa Clara County and San Francisco Bay to the north, Monterey Bay to the south. The valley is bounded on the east side by the Diablo Range, and to the west, the Santa Cruz Mountains. There's 5 major watersheds in the county, and Guad River and Coyote Creek drain to San Francisco Bay, while the Pajaro River drains to Monterey Bay. North County is pretty extensively urbanized, while South County is predominantly rural. The Santa Clara Valley Water District is a wholesale water supplier, and we serve 1.8 million people in the county. We have a trifold mission. It includes flood control, water supply, and environmental stewardship. We operate 10 reservoirs, and we manage over 800 miles of creeks and rivers. We also do other activities such as groundwater recharge, water releases, and diversions for fish passage. Our stream maintenance program for flood control covers the entire valley floor and the 5 major watersheds within the county. This is Anderson Reservoir. It's located in the east foothills above Morgan Hill. It was constructed in 1950, and it's the largest reservoir in the county, and the capacity of Anderson is to store 89,000 acre-feet of water. The Calaveras Fault lies within 2 kilometers of the dam, and seismic stability studies conducted in 2011 indicated that in the event of a 7.25 earthquake, the dam would liquefy. And within about 15 minutes, a wall of water 35 feet high could hit Morgan Hill and Gilroy, and then in a matter of hours extend north to cover San Jose as well. So the reservoir is currently restricted in the amount of water that we store in it, and we are engaging in a seismic retrofit of the dam. The price tag for this retrofit is approximately $200 million, and the project is scheduled to commence in either 2015 or 2016 and should be completed by about 2018. The dam itself bisects a ridge of serpentine soil here, and this is the very well-known Coyote Ridge just north of Anderson Dam. Coyote ceanothus, or Ceanothus ferrisiae, is a federally endangered shrub in the Rhamnaceae family. It's restricted to chaparral and scrub habitats on serpentine soils, and its populations are highly fragmented. Typically, it blooms from January through March. It's endemic to Santa Clara County. It's an obligate cedar, it's insect pollinated, and it depends on fire for regeneration. Coyote ceanothus is in the subgenus Cerastes, and it's most closely related to Ceanothus cuneatus bardubius, which is found at higher elevations in the Santa Cruz Mountains, usually on sedimentary substrates. As I mentioned, Coyote ceanothus has a highly restricted distribution. Historically, it's known from 4 putative populations, the Anderson Dam being counted as 2 occurrences within the CNDDB, or California Natural Diversity Database. There's a 5th historic occurrence in the Croy Canyon or Uvis Creek area from 1929. However, this occurrence has not been rediscovered, and it's possible that it's an erroneous occurrence. So currently, Coyote ceanothus occurs in 3 main areas in Morgan Hill. Here, this is the CNDDB mapped occurrence in yellow. So we have the largest occurrence at Anderson Dam, and the dam actually bisects this population, and it's the largest population at approximately 188,000 individuals. And in contrast, here we have the Kirby Canyon population, which is the smallest population. It's only about 150 individuals, and this population is not doing well at all. There's a lot of dead and senescent plants. Then you have the Yagas Road population, which is intermediate in size at approximately 1 to 2,000 individuals. And these populations are all separated from each other by approximately 1 to 3 miles. And Yagas is the most geographically isolated site.
What did the stars represent?
The stars were just sort of highlighting where these various population clusters are located. And then I should also mention that the blue and the brown are the serpentine soils layer as well as serpentine geology layer. I found that it's really important to look at both layers when you're looking at species that tend to be restricted to serpentine habitats. The district has several projects that will impact Coyote ceanothus. The largest is the seismic retrofit of Anderson Dam. Then we also have the reoperation of the reservoir. So currently it's kept at a restricted or reduced capacity. And we have ceanothus establishing along the shoreline of the reservoir. Once that reservoir becomes inundated again, those plants will probably not persist. And then we also have various dam maintenance projects that could also impact Coyote ceanothus. Impacts to Coyote ceanothus are covered under the Santa Clara Valley Habitat Plan, which is a multi-species, multi-agency plan with a 50-year permit term. Partners to the plan include the County of Santa Clara, the cities of Morgan Hill, Gilroy, and San Jose, and VTA, as well as the Water District. This plan proactively addresses conservation of covered rare and endangered species and natural communities while still permitting economic development. Biological advantages of the plan include the assemblage of a system of large landscape preserves and linkages, in perpetuity adaptive management and monitoring, and the stay-ahead provision, which basically says that mitigation has to stay ahead of impact. In addition, this plan is an NCCP, or a Natural Community Conservation Plan, which means that for species and habitats covered under the plan, there is a high bar for contributing to recovery of these species. So these two attributes, the contribution to recovery of a species and the stay-ahead provision, which says you have to conduct your mitigation prior to impact, gives us a really unique opportunity with regard to Coyote ceanothus, because typically you have a project and an impact and then you mitigate as a response to that project, whereas in this situation we have a lot of advanced lead time to really consider what an effective mitigation strategy would be and then ensure that that's carried out in the best possible way. So under the plan, impacts to Coyote ceanothus are limited to 3,650 individuals, or 5% of the population, whichever is less. And the plan says that a total of 5 occurrences will be protected in the permit area over the life of the plan. This includes the 3 known occurrences of Coyote ceanothus, and the Anderson Dam occurrence is counted as 1 occurrence rather than 2 populations. The plan also says that the mitigation strategy has to be implemented within 5 years of impact, and this is a little bit different. Typically the stay-ahead provision would say we have to have our mitigation in place prior to impact, but because we're on such an accelerated schedule with the dam retrofit, we have a little more time. So we can either preserve an existing population or create a new one. However, there's no known populations currently for sale, so our mitigation effort is focusing on creating a new population. This is an updated map of the location of Coyote ceanothus at Anderson Dam and Reservoir. And in this area here, this is a proposed retrofit of Anderson Dam. It's likely to change a little bit, but what you can see here is that the occurrence of Coyote ceanothus does overlap with our project area. This population, as the largest population of 188,000 individuals approximately, is so large because in 2003 there was a fire on this ridge here which is the southern end of Coyote Ridge, the population burned, and there was a significant recruitment since then, which has contributed to the large population size. So this is the timeline of our project. We've collected a series of baseline data. We've identified research priorities. We've engaged in a series of collaborative research partnerships. We're drafting our plan, conducting a feasibility study, and then based on the results of that and monitoring over a series of years, we'll finalize our plan, which will be basically a long-term management plan. And hopefully, if this all works out, by 2018, we'll have our full-scale planting effort of the new population. So tonight I'm going to talk about some of our field investigations and research studies, as well as how we selected a suitable introduction site. Our field investigations included soil sampling of known populations and potential introduction sites, habitat characterization, seed collection and seed banking at Rancho Santa Ana Botanic Garden, updated population counts within the existing populations, and field reconnaissance of potential mitigation sites. So I want to just make a little note here about habitat characterization, because Coyote ceanothus is a dominant or co-dominant in a community that's somewhat unusual because it includes bigberry manzanita, gray pine, and sagebrush. It's really more of a coastal scrub community, which is different than your typical chaparral in California that's dominated by chamise, manzanita, and ceanothus. In this case, Coyote ceanothus is a member of a community that's really more of a coastal sage, chaparral mix hybrid community, and there's not any component of chamise within these populations. And this habitat type is much more prevalent on the east side of the valley. So in selecting a potential mitigation site, our priority was finding a site with similar characteristics to the known populations. This includes similar soils and geology. As we know, Coyote ceanothus is restricted to serpentine soils, which is a pretty rare soil type. Similar elevation, slope, exposure, plant community associates, microclimate, and then other considerations such as land use history, habitat connectivity, and then climate change too is probably going to be very important. So in order to create a course level map of potential habitat for Coyote ceanothus, we took an overlay of several different data layers and created this map here. So we took the known elevational range of the species and the serpentine soils and geology layer from USGS, as well as the Valley Habitat Plan land cover types of serpentine bunchgrass, grassland, and serpentine chaparral. And then where all those layers intersected, we came up with this course level map of potential habitat within the county. So you can see here that most of the suitable habitat for Coyote ceanothus is on the east side of the valley along the Coyote Ridge area. The west side tends to have much less suitable habitat. The habitat there tends to be more mesic, it's higher elevation, more closed canopy, typical chaparral, and less suitable soils. In 2011, the district purchased this property here outlined in red. Now, we bought this property on Coyote Ridge in order to account for impacts under our stream maintenance program to red-legged frog and for stream and watershed protection. However, this is a great parcel because it also has numerous occurrences of rare and endangered species. It's critical habitat for bay checker spot butterfly, and there's quite a bit of serpentine soils on this parcel as well. And this property is located between two of the known occurrences of Coyote ceanothus. It's really only about a half a mile from each one of these populations. Now, on this whole entire parcel, there's actually not a whole lot of suitable habitat where we could create a new population of Coyote ceanothus because there's not a lot of serpentine chaparral. However, we did select this area here that we think is suitable. It has similar species associates, similar soils, slope, elevation, etc. And this area is approximately 22 acres in size. So meanwhile, we've also been working to learn more about Coyote ceanothus through a series of research studies. And there's questions that we're asking and trying to answer, such as how many populations are there really? Because the CNDDB counts the Anderson Dam occurrence as 2 populations. However, it's just bisected by the dam, so it would seem that it's all 1 population. And then once we figure out how many populations are there, how are these populations related? What do we need to know if we're going to create a new population? And then what do we need to do to make sure that this population is functional and self-sustaining in the long term? So we have a series of different studies. We have 2 related genetic studies. We have a water availability study, a soil interaction study, a nursery propagation study, and then our feasibility study on Coyote Ridge. So the 2 genetic studies, we have a Plastid DNA variation study that was conducted by Dr. Dan Potter's lab at UC Davis, as well as a microsatellite study conducted by Dr. Rodney Honeycutt at Pepperdine University. And these 2 studies were a partnership between the district, U.S. Fish and Wildlife Service, and these 2 public institutions. So the U.S. Fish and Wildlife Service and the district, we created the study design and the project plan. We went out and collected all the samples of plants, and then the district actually funded this work that was conducted by Davis and Pepperdine. And our overall goal was to investigate patterns of genetic variation within and between populations and to identify population structure. So we know that a series of genetic relationships among and within populations can help us guide and manage our conservation actions and hopefully contribute to the recovery of the species. And we also know that the loss of genetic variation and increased divergence within small populations can increase their risk of extinction. So I'm going to try not and be too technical here. This is basically a diagram of our Plastid DNA study results, and it's just a way to visualize the results. Basically, this diagram shows patterns of population divergence. So H is haplotype, and haplotypes are inherited ancestral genomic segments of DNA that change very, very slowly within populations over time. So as such, they're really great windows into species diversity, especially in populations that have been isolated for extended periods of time. So within Coyote ceanothus, we identified 4 different haplotypes. And in this diagram, each one of these lines represents 1 mutation, and the box size is proportional to the number of individuals that exhibit that particular haplotype. So you have H1 is found in all Anderson Dam individuals and some Kirby Canyon individuals, whereas H2 is found in other Kirby Canyon individuals and differs from H1 by 1 mutation. And then you have H3 and H4 were both found in the Yagas Road population. And they differ from each other by 1 mutation, but between 5 and 7 mutations from the other 2 populations. So basically what this is showing is that we have 2 major groups within the species, one with Anderson and Kirby and one with the Yagas Road population. There's some variation within the group, but the variation within is much less than the variation between the groups. And you have Anderson and Kirby, which are different yet related, whereas Yagas is really quite unique from the other 2 populations. This map shows the distribution of the haplotypes within the populations, and there's a lot of different samples that we took within the Anderson Dam area because we were really trying to understand, is this 1 or 2 populations? We also sampled the burned area as well as the unburned area to see if there was any difference there, and we didn't really find any. This ends up all being 1 population. So there are different population dispersal scenarios that you can look at with these haplotype results. You can look at whether dispersal occurred from the Anderson population originally through the Kirby population down to the Yagas Road population, or it could have been vice versa, where first you had the Yagas Road population dispersing towards the Kirby population and then the Anderson population. Another possible dispersal scenario was that the Kirby and the Anderson population on one hand and then the Yagas Road population on the other were founded independently by a common ancestor that's now extinct. The fact that there were H2 individuals found at the Kirby Canyon population but not the Anderson Dam population suggests that either one of those two populations was the source of the other. And this is really interesting because we know that in 1992 there were propagules taken from the Anderson Dam population and established at the Kirby population because the Kirby population has always done very, very poorly, and there was an attempt to restore that population using Anderson Dam propagules, and that's probably why they have that shared haplotype. So just to summarize, we know that the distribution of the major haplotype variation is correlated with the geographic distance and location among populations. We know that there's an east race and a west race. Within each race, there's two subraces, and the genetic divergence between the eastern and the western populations indicated that they've been separated from each other for a relatively long period of time. So we also had a DNA microsatellite study, and in that study we were addressing 4 main questions with regard to population structure. We wanted to know how many distinct putative populations exist within the species, and based on geographic distance, is the Yagas population the most divergent? Are the existing number of individuals at each site a good predictor of levels of genetic variation? And is there an indication generally of a loss of genetic variation within the species? So Dr. Honeycutt used a panel of DNA microsatellite loci to investigate patterns of genetic variation within and between populations. And before this study, there was no microsatellites developed for Coyote ceanothus specifically. So Dr. Honeycutt actually used primers for a related species that he worked on down in Southern California, Ceanothus megacarpus. So the results, I won't go into all the different statistics that they do in microsatellite analysis, but basically it confirmed results from the Plastid DNA study that we have 3 main populations. You have one at Anderson Dam, one at Kirby Canyon, and one at Yagas Road. So genetic diversity can be estimated based on number of private alleles per locus, average number of private alleles, and estimates of heterozygosity. And the main thing to really take away from this table of the results here is there's major differences with the Yagas Road population and the Kirby Canyon population. In all cases, the Yagas Road population had the highest number of alleles per locus, the highest number of private alleles, and the highest levels of heterozygosity, indicating that this is the most genetically diverse population of the 3. In contrast, the Kirby Canyon population had the overall lowest levels of genetic variation, meaning that it is the most ungenetically diverse of all 3, whereas all these different areas of Anderson Dam were somewhat intermediate between the 2. What this microsatellite study also showed us is that these levels of genetic variation are really, really low, considerably lower than what we've seen in other species of Ceanothus. In fact, only Ceanothus arboreus, which is an island endemic in Southern California that exists in highly fragmented populations, only Ceanothus arboreus has similarly low levels of genetic variation compared to Coyote ceanothus. So what is this really telling us, these studies? Well, we have an east and a west race, which seems logical based on the geography and ecology as well as the genetics. Geographically, the Yagas population is located much farther west and closer to the Mesic-Santa Cruz Mountains, while the other 2 populations are located across the valley floor in the east and inland Hamilton Range foothills. The unique haplotypes in the west race, coupled with the microsatellite results, indicate the Yagus population is definitely both distinct and more heterozygous. And the number of mutations also suggests that Yagus has been isolated for a very, very long time. So what this could be telling us is that Yagus is actually the origin population for the species, and that it's actually a relic population of a once more widespread distribution of Coyote ceanothus. Kirby possibly could be a peripheral population of Anderson that's become more isolated over time, which makes sense because, as I mentioned earlier, the population is actually doing very, very poorly with a lot of dead and senescing plants. So our propagation efforts and ultimately a successful creation can be really influenced by the propagules we choose. We can actually collect propagules from specific individuals that we know have higher levels of genetic diversity. And since we know that Kirby Canyon has lower levels of genetic diversity, then we can actually prioritize this population for a recovery effort. So the next question we asked, I showed this map earlier. If you have what seems like quite a bit of suitable habitat, why is Coyote ceanothus so restricted, and why have we only found these 3 populations all localized within a relatively small area of Morgan Hill?
How is it pollinated?
By bees and insects.
And so they probably don't move between populations?
Probably not, depending on their dispersal distance. So, is the species dispersal limited? Perhaps there's a long fire return interval, depending on the fire pulses within the existing population areas. It's possible that there's just not enough fires to promote recruitment. So we also wondered about the role of microclimate in distribution of these populations. California is a Mediterranean type climate, and it's somewhat unique in that you have a summer dry season that's moderated along the coast by a persistent layer of fog and cloud cover. In past work done by Dr. Mike Vasey, he found that the summer marine layer at a regional scale can really significantly enhance the availability of water availability for upland coastal shrubs compared to interior chaparral shrubs. And he also found that the highest level of local endemism in the Central Coast region can be correlated to the availability of water when you're on a gradient from the coast to the interior conditions. And what I've seen in my work in South County in the Morgan Hill area is that you really do get a very obvious marine layer that comes along the Pajaro River, it flows through the Pajaro Gap, and then it's very localized around Morgan Hill. And then by about lunchtime, this whole fog layer burns off, and then it gets really hot and humid, and the area is actually very arid. So we hypothesized that Coyote ceanothus could be a local endemic distributed within this summer marine layer zone. Dr. Vasey also found that relatively small elevational changes can potentially result in large differences in the influence of the summer marine layer on vegetation composition. Coyote Ridge, our potential introduction site that we preliminarily selected, is kind of a little bit higher in elevation than the general range of the known populations of Coyote ceanothus. And this picture was actually taken from the top of the ridge, basically where our potential mitigation site is located. And you can see this thick fog layer that's covering the valley and Morgan Hill below. So we have this marine layer. It comes down through the Monterey Bay, moves inland through the Pajaro Gap. However, Morgan Hill is really only a short distance from truly interior conditions. So we could really be on the boundary here with Coyote ceanothus. So this is something that we really wanted to study. We thought it was important in selecting a potential introduction site. So we asked a series of research questions. Is the water availability from a combination of the summer marine layer and the winter rain a factor that does constrain the distribution of Coyote ceanothus? And how important are these climate variables in the selection of a suitable introduction site? This is Mike setting up his research within one of our sites on Coyote Ridge. Again, lots of fog. So Dr. Vasey conducted a study of dry season water availability in the existing populations of Coyote ceanothus, as well as a microclimate suitability study of the potential introduction site located here on the ridge. And although this introduction site appears suitable, based on the exposure, the plant species associates, and soil factors, its increased elevation over the known populations of Coyote ceanothus really raised the question as to whether its water availability would fall within the range of the species. So we installed localized weather stations in all 3 known populations of Coyote ceanothus, as well as at our potential mitigation site. And we measured temperature, relative humidity, leaf wetness, soil moisture, and precipitation over the period of 1 year. We also conducted late dry season water potentials to estimate comparative drought stress in all the populations of Coyote ceanothus. And we were actually also able to access a portion of the Anderson Dam occurrence that's located on Coyote Ridge. It's at the highest elevation for that population. It's on private property, and it's actually quite close to our potential mitigation site. And then we also downloaded some long-term climate data for comparison. So basically, this graph is showing comparative drought stress, where these are water potentials, and this represents extreme drought stress, and this represents no drought stress at all. So water potential as a measure of drought stress. And in these different box plots here, this is Anderson Dam here. This is the August Road population. These were 2 different exposures within the Kirby Canyon population. And then this was the top of the Anderson Dam population on Coyote Ridge. So we were really surprised to find that the Anderson Dam, especially in the post-burn area, these plants are actually the most drought stressed of all the different populations. We didn't expect that, but what we think is happening here is that there's some density-dependent thinning going on. There was so much recruitment post-fire, you have a lot of plants in a very small area all competing with each other. So, and then this was surprising too, the Kirby populations, because we were thinking, well, maybe, the Kirby population's not doing so well, maybe it's a drought stress. Well, this proves that that theory is completely wrong. Then you have Yagas, intermediate, and then Coyote Ridge actually intermediate as well, which is great, because what this is telling us is that that introduction site probably is suitable as far as a location for us to create a new population. So these values generally are pretty low. However, they're not as low as other comparative species of ceanothus in the literature. So what this is really telling us is that generally Coyote ceanothus is not terribly drought stressed, and so that indicates, in addition with our meteorological station data, indicates that there probably is an influence from the summer marine layer that's contributing to the hydrology of the species and keeping it from getting too drought stressed. But then you have some portions of the population that are really drought stressed, which tells us that the species can withstand some drought stress.
Is that just a single year's data?
That was a single year's data. So we also looked at soils, and this was a study that we did with Dr. Alison at UC Davis. And she was looking at Frankia, which is actually a soil bacteria that associates with species of Ceanothus, and it causes the species to form nitrogen-fixing root nodules, and up to 60% of nitrogen, soil nitrogen, can be uptaken by the plant through this method. There hadn't been a whole lot of research to date on whether Frankia existed in serpentine soils because they're so dry and nutrient poor. And then if it does exist, we didn't really know if it formed an association with COYOTE ceanothus. So this was the purpose of this study, was trying to identify whether Frankia was found in existing populations and whether the plants actually form root nodules. And we did confirm these are root nodules of Frankia on COYOTE ceanothus at Anderson Dam. So our methods, we used a stable and isotope analysis of leaf tissue samples that were collected, as well as extraction of DNA from soil samples. So between those two, we were able to get these results. So then we're asking, well, is this important when we plant our seedlings into our potential mitigation site? We did identify positively Frankia in soil samples from the two populations we tested. Some of these sites have a really weak signature, and some of these Frankia strains are actually identical to strains that were collected way far north in California at Sage Hen Creek Field Station, which indicates that Frankia in general is probably, it's not a super specialized strain within one small area of the state, but it's actually generalized across the state. But we did not identify any Frankia in soils collected from our Covole Ridge potential introduction site. And the way we collected the soil here is you carefully excavate soil from around the roots of a mature ceanothus plant, and from a depth of approximately 4 to 8 inches, and then you just replace the topsoil, and it's a non-invasive way of collecting soil. So we know that we have a positive idea of Frankia within our existing populations. We know that we have no Frankia in our introduction site, so that tells us that we have to inoculate our seedlings that we're going to plant into the introduction site in order to ensure a higher chance of success and in order to help those plants fix nitrogen. So that's what we did. I collected seed from Anderson Dam. We grew it in a nursery. We inoculated the seedlings. Now, one of the things that we found out is that the success of the inoculation is really dependent on the amount of soil that you add and the good contact with the root mass. And our first attempt at inoculation, we only had about 10% of our seedlings inoculate, but it was really, really interesting because the ones that did successfully inoculate looked way bigger, they're more vigorous, they're darker green. It might be a little hard to tell in this picture, but this flat were all seedlings that successfully inoculated, and these other seedlings did not successfully inoculate. So what we did during planting time was add more inoculum. So this is our feasibility study on Coyote Ridge, and we actually just finished planting our seedlings this week before the rain. We have 4 test plots in different habitats located on the site. So we have an upper sagebrush test plot and a lower sagebrush test plot, and this is to take advantage of any elevation difference and potential fog microclimate difference on the site. Then we also have a test plot that's within a sort of a gray pine overstory grassland area, and then another test plot that is located at the edge of an existing serpentine chaparral area. So there's not much serpentine chaparral on the site, and the chaparral that is there is pretty occupied, and we didn't want to go pulling plants out and inserting new plants in. So what we did was just kind of extend that chaparral habitat out into the grassland. And so the purpose of these test plots really is to see if there's any difference in planting or incorporating Coyote ceanothus within these existing little different species associates or communities, because what we really want is a functional population over the long time. We don't want a garden of just monospecific Coyote ceanothus. We want a functional community, and we're not totally clear what other species make up that functional community. We looked at the existing populations, and we're trying to use that as our model. So these seedlings will be monitored for 4 years, and hopefully at that time we'll be able to tell whether this creation effort will be successful. So just to wrap it all up, what do we know so far? We know there's 3 genetically distinct populations in 2 races, an east race and a west race. We know that the Kirby population is more homozygous, and that population appears to be in decline, but not from drought stress. We know that Yagus is the genetically robust and most distinct population, and that Anderson is moderate in its levels of genetic diversity, with some evidence of post-density dependent stress in the burned portion of the population. We know that in general, Coyote ceanothus has relatively low levels of genetic diversity compared to some other species of ceanothus in the literature. And we know that Coyote ceanothus is fairly well hydrated compared to other species. Therefore, there does appear to be an influence from a marine layer. The potential introduction site on Coyote Ridge appears suitable. We know that mature shrubs of Coyote ceanothus can tolerate a lot of drought stress, but the Coyote Ridge site appears to be within the limits of the species, and our meteorological station data confirm this. We know that our introduction site will benefit from the inoculation of Frankia for the establishment phase, and we know that data collected can guide our selection of specific mothers for propagation. So we can combine the results of the genetic studies with information about the physiological tolerance of individuals within the population in order to select specific shrubs for propagation that might be better adapted to our introduction site. We know that fog is super important. Another thing from a pure genetic standpoint, if we want the highest levels of genetic diversity, maybe we should be mixing all these populations together. Maybe we should go to the Yagus population. If that's the most genetically diverse, should we go there and collect our propagules from the Yagus population? But, you know, if there is some sort of microclimate adaptation on the east side of the valley, then perhaps bringing the west side over isn't such a good idea. But these are the kinds of questions that we really have to ask ourselves, especially in the case of climate change. So this is Anderson Dam, the post-burn population, lots and lots of plants in a very small area. This is the O'Connell property, which is the northernmost portion of the Anderson Dam population on top of Coyote Ridge. And really, this is what we're going for, is something like this, a functional population, a variety of different community associates that hopefully will persist over a long period of time with minimal interaction from us. This is after the burn. The burn was 2003. This was the first year, 2004. Lots of recruitment, lots of open areas, all the pines burned. And this is one of my favorite shrubs at Anderson Dam. It's always absolutely covered with blossoms. It's quite showy. It smells wonderful. So really what we need to kind of remember in looking at this slide is that fire really is a part of the process of regeneration, and that Coyote ceanothus could be a fog-influenced community driven by fire regime. And we need to really explore what type of fire regime Coyote Ridge has historically experienced and try and incorporate that into our long-term management of the population. A lot of different people have contributed wonderful, amazing ideas to this study, and this has really been a wonderful, wonderful opportunity to partner. And there's just been a lot of great things that have come of this. I want to acknowledge all these people and just say it's an ongoing dialogue, it's an iterative process, and there's really always more to learn.